Hello, everyone. Today, I'm going to quickly talk about some machine learning software and how it's being used in environmental studies and what it can do for research. All right, so for those of you who don't do big data or don't do machine learning a lot, basically what it's doing right now is it's driving some pretty cool innovative approaches to LCMS data analysis. Most of this presentation, we're going to focus on high resolution accurate mass data. So think instruments such as a QTOF or a TOF or an Orbitrap, and not really triple quad or single quad instruments here. So on the left here, we have a picture of a type of machine learning algorithm called a neural network. And basically, what we're doing here is MS Omics data sets are large and highly complex, as well as mass spectrometry work data sets in the environmental realm now as well, especially non-targeted analysis, sometimes suspect screening. So really new analysis techniques are required. You can't really do everything manually with human eyes anymore. And machine learning techniques are really kind of leading the charge here. And I'm hopefully going to try and convince that of you a bit during this presentation. There are a couple different types of machine learning techniques that can be utilized. And we actually make some software, as well as some of our competitors, that can do this well. So one of the first softwares that were kind of ever to implement machine learning algorithms and mass spec data is something called Mass Profile Professional. And that's something that Agilent Technologies makes. It can interpret almost any type of mass spectral data, GC, LC, ICP, as well as metadata, as well as third-party data too. So it doesn't need to just be Agilent instrumentation. And it's very, very advanced statistically. So we have a lot of statistical tools in there and powerful visualization packages. So on the right here is a visualization of a random forest variable importance model. On the right here, we have something called classifier. So this lets you build class prediction models in that software as well, which can help authenticate or classify your samples. And I'll talk about that a bit later and what it can do. This is very easy to use, right? So there's a node-based workflows, drag and drop what you want to do in the software. So it takes kind of the coding part out of machine learning analysis for you. So you don't need to be an expert coder or sometimes even an expert stats person in order to do some of this. So I talked about earlier saying there's two different types of machine learning models. That's unsupervised and supervised. Basically, in unsupervised, you're going to correlate patterns of abundances across samples without really having an understanding of the sample groups or deciding to be blind to them in an algorithmic sense. So here, think of a principal component analysis or a hierarchical cluster analysis. This is often done a lot of times when you have big data. So some of you might be familiar with these terms. Supervised means you're going to use sample grouping to find patterns and abundances and discriminate groups with more power. So think PLSDA, think random forest, think SIMCA. So this can indicate what compounds are most important to your study, say if you're doing a case versus control or authentic versus non-authentic. You can also analyze your unknown separately from your model samples. This doesn't really, it's not a panacea, right? It's not a cure-all. You still need basic statistical approaches for good supervised models, right? So you can't make good models if all you're looking at is noise or all you're looking at are insignificant features. So you do need some simple stats understanding in order to filter through your samples, especially when you're looking at big data and non-targeted analysis. So you want to look at detection frequency across your replicate injections if you have them. You want to look at CVs or coefficient of variations, meaning you want low variability in a group. You want to maybe do some fold change analysis, some blank subtractions, that is. A t-test maybe. Features should have large differences between your groups in your machine learning models if possible. And ideally, even an even number of features in different groups is even better. So on the right here, we have some apples and comparisons between different apple types. So we have the number of compounds that are significantly different in comparison highlighted versus the number of compounds that aren't significantly different. And you can see between the different varieties of these apples, there is quite a bit of significant difference. So how do we implement this in a workflow? So for a small molecule machine learning workflow, we use what we called earlier called classifier, which is in the MPP package. And basically we could put data from our samples in there. Say if we're working with mangoes, we could also do what we just did with apples. We can take the different varieties of mango, put them in, all the features we detected in each one, and it'll basically spit out a PCA showing us what features compromise specifically those types of mangoes. And here on the right, that's what we're looking at. We can see that there are different cluster groups for each type of mango. And from that, we can start to predict what kind of mango we have based on an unknown sample and get a certain confidence amount out of it based on where it sits. So I just talked about a bit about machine learning and classification and how it works and where it works. Now I'm going to touch on for a second. So we're in environmental mostly here, so I'm going to focus on that. So what can this do for an environmental work? You could do source apportionment. So where are contaminants coming from and can we predict it quickly? Routine monitoring as well. Is my sample safe or do I need to act now to prevent environmental consequences? So this is also important though in food analysis, right? Food analysis and environmental analysis, I find kind of can go hand in hand, right? So authenticity, is this product what it claims to be? Is it the right composition and from the correct location? Does my product meet the complex chemical profiles that consumers expect for quality? It also has uses in life sciences and biopharma, but we could skip over that. So a little quick example of how this might work in environmental fashion. This is a real project. So endangered rosewood is a part of illegal logging operations. So we wanted to see if classifier could find unique markers that could be used by customs for testing shipments, right? Remember, we want good statistical practices, good sample prep. So we did 10 plus replicates per group. Unidentified features were significant between species, and classifier identified unknown species correctly. And we used basically a random forest model for variable importance that we saw earlier. And we also looked at a PCDA or a PLSDA and PCA. And classifier basically identified unknown species correctly. We have a suspect sample that's highlighted here versus all the different types of real rosewood. And what it was able to do is it was able to correctly identify the unknown species that were taken from illegal logging operations. So basically, this is all kind of under the chemometrics umbrella. And hopefully this can show that chemometrics can improve your lab's research. We have software to help with big data. We can improve various products. And basically, proper hardware, experimental design are key to the meaningful data in machine learning technologies, and scientific expertise that we have is available and within reach. And we can get you started on this research if you're interested. A couple quick acknowledgements, and thank you for having me come present. I'll take any questions if we have some time.
We do have a couple minutes. Are there any questions? Thank you very much.
I don't see any at the moment.
We'll wait a minute and see if anything pops up.
All right, I see one.
Oh, you see it.
Yeah, all right. So the question was, do these workflows work with Thermo products? Yes. So as I said, we can take in kind of, so we're kind of one of the only vendors with a platform that has machine learning capabilities, but we can take in data from any of the other vendors and use it in these platforms. All you really need to do is it needs to be able to go into kind of a CSV format and we can take it in. Yeah, it's not difficult.
Great. Any other questions?
I checked Tuva and I don't see it. Oh, there's one more. Can I identify compounds in libraries or not in libraries? Can identify compounds not in libraries?
So yeah, so we can do formula generation that's not based on a library. We could also do MSMS work to get structure elucidation when you don't have a library. we could also do it with a library as well. We can match to a library if necessary.